Yeah, so this is hemoglobin. Anybody tell me the structure of hemoglobin as far? Well, yeah, heme is, what would heme be called then? So it's this prosthetic group, right, with iron. But there's 4 proteins that make up hemoglobin. There's, you have 2 alpha hemoglobin molecules and 2 beta hemoglobin molecules. So it forms, to have the proper quaternary structure of hemoglobin, you have to have all 4 subunits together. A lot of proteins are like that, where there's multiple subunits that make the overall protein and give it its function. So that's the quaternary structure. What were the other levels of structure that we talked about last time? Yeah, so what are those? What's the primary structure?
Sequence of amino acids.
Exactly. Secondary structure, local 3-dimensional structure that makes up like your alpha helices and your beta pleated sheets. And what stabilizes those alpha helices and beta pleated sheets for the most part?
Hydrogen bonds.
Yeah, hydrogen bonds between what?
Hydrogen bonds between sheets.
Yeah, but is it the R groups that's forming the hydrogen bonds, or is it the peptide bond atoms?
The oxygens.
These oxygens and the nitrogens are actually the hydrogen donating part, right? So it's the peptide bond atoms, the nitrogen in that amide bond and the carbonyl oxygen in the carboxylic acid of that amide bond. It's those that form the local secondary structure. So don't confuse it with the R groups. The R group is going to help form the overall tertiary structure, how that protein folds, right? Because if you have hydrophilic residues, they're going to tend to be exposed to the solvent in the outside, just like hemoglobin. And then on the inside, you're going to have more hydrophobic molecules. So how do they come up with these? Any of you guys read the book yet? How do you determine protein structure, quaternary structure of a protein? Because they're way too small just to look at, right? So how do you do it? Well, yeah, you have to purify it really well to do either method. But there's X-ray crystallography. Have you guys ever heard of that? So basically, you get the protein as pure as possible, then you make a crystal with it. And then they'll hit it with X-rays and look at the way that the X-rays diffract, because the X-rays aren't going to be able to go through the nuclei of those, all the atoms in the proteins, and there'll be a diffraction from those nuclei, and you'll be able to mathematically put together the structure. That's how they determine the DNA structure too, right? What was her name? Rosalind Franklin? Yeah, she actually did the X-ray crystallography of DNA, but hadn't determined the structure. And then Watson and Crick, I guess, saw her images and kind of stole her data to come up with the structure of the double helix. But anyways, so the steps needed: you have to purify the protein, crystallize it, and that's a tricky step. Collect the diffraction data, and then calculate the electron density from that diffraction data, and then you fit residues into the density, and it's a long, arduous process. Pros is there's no size limits to the size of protein you can examine that way, and it's well established. That's how they established the structure of DNA. Cons is it's difficult for membrane proteins, and you cannot see the hydrogen. Why would it be difficult for membrane proteins? Well, because for them to have their proper secondary structure, they have to be in a membrane which is kind of hydrophobic, right? So you have to have some sort of hydrophobic solvent to crystallize them out of, basically. Does that make sense? So they're not easy to crystallize where you're just evaporating the solvent off, like water you would with a normal aqueous solution. And this is the kind of data you get. This is the X-ray diffraction, and then you can kind of piece together the electron density, and with the computer determine the structures, and eventually get these ribbon diagrams. And these are all, do you know what the ribbons stand for? Here?
Those are also?
Yeah, alpha helices. So Another method actually worked in a lab that did this was NMR. So they take proteins and determine their structure via NMR. And again, you have to get the protein super pure, then you have to dissolve the protein in the solvent. And the lab I work in actually would do membrane proteins. So they would create micelles to have the membranes, redissolved in the micelles. Collect NMR data and then assign NMR signals and calculate the structures. Pros, no need to crystallize the protein, which can be a real hassle. And then you can see the hydrogens. And the cons, it's difficult for insoluble proteins, even though the lab I worked in would use membrane proteins. And then it works best with small proteins. Why would you have to have small proteins? Anybody know how NMR works? So basically, you align all the. Well, it depends on what type of NMR. There's proton NMR, C13 NMR. I think it's N15 NMR. Number of different isotopes you can look at. But basically, you hit it with the magnet, and it aligns all the paramagnetic atoms, and then you let it go back. They'll all spin the same direction, basically, and then when you take that magnetic field away, they'll drop back to their ground state and release radio energy while they fall back to their normal spin. So you line them all up and then measure basically the anisotropy. Basically how long it takes them to go back to their normal spin.
That's the best mode of measuring can be NMRs.
Well,
You don't have to crystallize it.
you don't have to crystallize it. It doesn't necessarily mean it's the best, but one of the advantages actually to NMR is that you can actually look at multiple conformations in one tube. Like you can do an NMR cycle with a protein, then add some kind of ligand and look at a conformational change. Or most conformations actually are in equilibrium between the protein having one shape and then it'll exist in the other shape temporarily and go back and forth, and there's an equilibrium between that. And when you add a ligand, it shifts that equilibrium one direction or the other. So you can pick up both conformations normally in NMR, and without the ligand, you can look at the equilibrium constants with that. And this just shows you like a 2D NMR structure and how that looks like. You're looking at two H1 chemical shifts, which should be a perfect line like that, and you can look at differences between the two by measuring those. And then you'll get structures like this because there's movement a lot in NMR, unlike crystals. So you get like the how much is, I forget what that's called. Family structures consistent with the distance constraints to the NMR. Not only can you do that, but you can put non-paramagnetic isotopes on particular molecules to help you identify which amino acid is where when you're piecing together NMR data. So intrinsically disordered proteins, and so there's some proteins where there's sections of them that are just intrinsically disordered. And they contain protein segments that lack definable structure, composed of amino acids whose higher concentration forces less defined structures. Didn't I give you the example of a polylysine protein? It's going to be disordered because of all the charge repulsion. Because what charge is lysine at pH 7?
It's neutral.
No. Basic, yeah, so it's a positively charged. It grabs a proton from water, gives it a positive charge, so it's going to be positively charged at pH 7. If you have a bunch of polylysines there, they're not going to form alpha helical structure because they're going to repel each other and they won't interact with each other. Same with arginine, glutamic acid, and proline doesn't like to form alpha helices to begin with. Because it's why is that? Yeah, what about its structure makes it not want to form like alpha helices? Those psi and phi angles are pretty confined because if you remember, proline is that ring structure. It's the one that's cyclic. So. Yeah, it can exist in that cis or trans conformation. I think we talked about last time, but you can't get rotation around the psi or 5 bonds like you can with basically all the other amino acids. So disordered regions, they can conform to many different proteins, facilitating interaction with numerous different partner proteins. And I think the example they have here for that is, what is this again? So it's basically this P53 protein. Have you guys heard of that? Taking immunology or... So P53 you ever heard of oncogenes? Mm-hmm. Yeah, P53 is discovered because it's an oncogene. And so what do oncogenes do typically? What are they involved with? Yeah, so typically they're involved in regulating cell cycle, and that's P53 is also. So it'll bind to a number of other proteins, and it's this, if you look, this is a Ponder score, which basically scores the order or disorder in the protein. So they go down through the amino acid residues. Everybody remember a residue is a single amino acid. You start at 0. At which terminus, Dina?
CO.
The N terminus. So 0 is always the N terminus to the C terminus. So here we have the N terminus, and it's actually highlighted here. That's fairly unfolded, but then I think it's the C terminus is involved in all these binding domains. Cyclin A, so cyclin A is a protein that regulates cell cycle. Sirtuin, CBP, bromodomain, and then the S100 beta, or beta. So, proteostasis, have anybody ever heard that term before?
Isn't like Barmo, it's like in the same group as like Barmo, oncogenesis, like kind of like proteosis. It's like
It's a proteostasis, so it's like basically proteo is like for proteins, and stasis is what?
So.
Yeah, so it's like maintaining the status quo of your proteins in your cell. So basically in proteostasis is the maintenance of cellular protein activity, and it's accomplished by the coordination of a family of different pathways. So you have your nascent protein being produced, and that's got to fold properly. So and a lot of times you have folding intermediates based on the free energy diagram of how it's going to fold. Sometimes folding is going to require certain proteins known as chaperones. And what they'll do is they'll bind to the growing nascent protein as it's starting to fold itself, and they'll make sure it maintains its native fold structure, and also protect certain regions from interacting with other proteins. Because if you just produce proteins as they come out, it's real easy for hydrophobic regions just to start forming cross beta sheets, and they'll just fall out of solution and form like amyloid fibrils. So you have a native protein fold that eventually will get broken down into peptides and fragments, or it can unfold and you can have a misfolded protein which will be signaled for degradation via the ubiquitin proteasome system. Have you guys ever heard of ubiquitin? It's called that because it's ubiquitous. It's found ubiquitously in your cells. And it's basically a protein that gets added to bad proteins, or proteins that are marked for destruction, basically. You get ubiquitin, multiple ubiquitin proteins are covalently attached to a protein, then it's subject to degradation, and it actually will go to this big trash can protein that'll break it down into little fragments, the proteasome. So folding intermediate, you can, if it unfolds, it can be refolded via chaperones, or you can get disaggregation back to the folding intermediate, or aggregation, which if you guys heard of like the amyloid disorders, so Alzheimer's, Parkinson's, prion disease, you can even get amyloid buildup in your pancreas with diabetes. I can't think. There's inherited forms of diabetes where you form amyloid particles of insulin in your pancreas. Anyways, what happens with those is you get the cross beta sheet structure between proteins, and they'll just fall right out of solution. Or you can have oligomeric structures, which with Alzheimer's they theorize it's these oligomeric structures that are actually the toxic amyloid, or amorphous aggregates. Autophagy is one way that they clear this. It's a way that proteins are broken down, where they, you know, what are phagocytes? They take in and engulf something and digest them. Autophagy is where it kind of does that on its own. So protein stability and folding. Protein's function is dependent on its 3D structure, right? So if you don't have the right 3D structure, it's not going to bind to its particular ligands. It won't be able to catalyze reactions because those reactivities are all governed by this amino acid being here, stabilizing some transition state, and so forth. Loss of that structural integrity is accompanied with accompanying loss of activity is called denaturation, and well, I guess I have it up there. Like how do you denature proteins? Denature proteins is just unfolding them. One way is just by heating them up, right? So you heat them up, you're going to add energy into that protein, and you'll be able to break those hydrogen bonds that are stabilizing that secondary structure, and you'll get more free rotation around all the other bonds, and you can take apart that protein. Although which covalent bonds doesn't like to have rotation around? Can you tell me? Not the double, well, yeah, double bonds don't, but what is it about proteins that doesn't like to rotate? Yeah, the peptide bonds, because it has that partial double bond character, right? Why would pH extremes unfold a protein? So pH extremes are going to unfold it because you're forming all these hydrogen bonds. Those hydrogen bonds are between ionizable groups. So that nitrogen that's donating that hydrogen to form a hydrogen bond with that carbonyl oxygen, right? If you lower the pH, that oxygen may pick up a protein. You can no longer form the hydrogen bonds between the two. So that's one way you're going to unfold it. Organic solvents will unfold proteins because they're going to allow those hydrophobic residues to be exposed to the solvent, rather than hydrophobic forces are one of the main driving forces. It's the hydrophobic force is one of the main driving forces for folding proteins. And then, I don't know how to say this, chaotropic agents. Have you guys heard of that? Chaotropic. So these are agents, like if you look at the structure of urea and guanidinium, there's a lot of NH there. So basically you're going to saturate any of those hydrogen bonds that you get to form the secondary structure with the nitrogens or the carbonyl and urea and guanidinium. And this is actually one of the ways I've used to extract amyloid deposits from the brains of mice inflicted with human amyloidosis.
Does naturation and denaturation of the peptides occur because of the specific organic additions, or is it just the solutions that surround it?
The denaturation or renaturation?
Denaturation, renaturation of peptides. Is it a solution variable, or is it?
It can be a solution variable, yes. Like if you have like 5 molar urea, it'll dissolve almost any protein.
Okay.
So, and that's because the structure of urea has those 2 amine groups and then a carbonyl, and those will like basically disrupt the hydrogen bonding characteristics of that secondary structure of a protein and unfold it. And then if you take that denatured protein that's in a high concentration of urea, you can dialyze the urea away, and that protein will return to its native fold for the most part. Okay. So, and this is just an example of protein denaturization, showing there's specific temperature the proteins are going to unfold at. They use ribonuclease A because it's an easy one to test, because you can basically breaks down ribonucleases like RNA, and so you can measure the activity at different temperatures, and you can see you lose activity as you increase above. Basically, the melting point is what, about 50 degrees or so. And same thing with apomyoglobin. So a lot of proteins are going to have different denaturation points. So, and then this shows you denaturation of ribonuclease A as the increased concentration of guanidinium hydrogen chloride, which that's what I use to take apart amyloid plaques. So they did this experiment, ribonuclease refolding experiment. And ribonuclease is a small protein that contains 8 cysteines linked via 4 disulfide chains. So these have disulfide structure. So if you think about it, you unfold it and break those disulfide bonds. Why would it refold the exact way? Wouldn't it just make different new disulfide bonds? So what they did is they added urea in the presence of 2-mercaptoethanol. You guys know what the 2-mercaptoethanol is, or beta-mercaptoethanol? Yeah. It's that stinky stuff, right? Huh? Yeah, so that beta-mercaptoethanol, you know, it's got a sulfhydryl group on it, and it'll basically react with the disulfide bonds and reduce those back to sulfhydryl groups. Okay? So it's used to cleave all the disulfide bonds. So when you do a Western blot or a page gel, you typically boil your protein to unfold it with 2-mercaptoethanol to break any disulfide bonds, to break apart the different peptides making up the quaternary structure of that protein. And when the urea and 2-mercaptoethanol are removed, and they do this via dialysis typically, the protein will spontaneously refold, and then the correct disulfide bonds are reformed, and the sequence alone is what determines that native conformation. So it's a real simple experiment, and it was done way back when I was born. So, and this is basically it. So the native state, you have particular disulfide bonds, you unfold it with the high concentration, they use urea or guanidinium. I think they used urea. With beta-mercaptoethanol, reduces the thiol groups. And then we remove the urea and the beta-mercaptoethanol, and it reforms the exact same structure. So it's that primary sequence that governs basically the structure, the quaternary structure of the protein. And they do this, a lot of them are going to follow a particular path. So there'll be localized structure that'll start to form, let's say for this example, the residues 30 through 37 start with an alpha helices, and it just kind of falls from there down the, it's free energy, how it's going to do that. Same thing with the beta sheet structures as they come together, and eventually they'll fall down to the lowest energy state, and that's typically going to be that folded protein. So proteins fold to the lowest energy fold in the microsecond to second time scales, and it's mathematically impossible for protein folding to occur by randomly trying every conformation until the lowest energy one is found, right? So they just naturally are going to fall down towards that lowest energy state. And the search for the minimum is not random because direction towards its native state is thermodynamically favored. And they have all these like graphs basically of how proteins will fold, right? So, and this is in free energy, so this would be the lowest state or the natively folded state. You can start anywhere along here and it'll always end up in its lowest state. But most proteins are going to fold, will look something like this, where we have these intermediates that you have to like little humps that you're going to overcome before you get to the native state. So. And chaperones, did I tell you about chaperones already? They're basically proteins that are going to help chaperone the protein and help them fold to their native state. And they'll help prevent aggregates because as that protein's coming off the ribosome, like I said, it's real easy for it to interact with other unfolded proteins to form these cross beta sheet structures, and they'll fall right out of solution. So chaperones were discovered, and they'll bind to these and just stabilize the unfolded sections until it can interact with itself and fold its native protein. Some require ATP to bind and then to come off. Yes?
What are aggregates?
Aggregates is basically where you have proteins that they'll precipitate with themselves out of solution. So that's what I was saying. They'll form these cross beta sheets. This is a beta sheet structure. If you form cross beta sheets with other proteins, they'll tend to start like a chain reaction. They'll just drop out of solution and form the aggregates. So there's one group of proteins called chaperonins, and basically they're found in E. coli. The GRO-EL or GRO-EL, it's basically like a folding machine. So the unfolded protein will go in, and it's an ATP-dependent process. The protein binds to the side, and it'll every time it binds to the inside of this chaperonin, it'll fold a little bit more and burn an ATP with each binding event. And some proteins can take like several hundred ATP before they're ejected in their folded state. And that's the crystal structure of a chaperonin right there. So it's quite a large molecule. Protein misfolding is the basis of numerous human diseases. And this is what I was talking about, where partially misfolded protein here, if you notice the beta sheet structures, and then they'll associate with other beta sheet structures and other misfolded proteins and just form these filaments of amyloid fibers where they just drop right out of solution. And this is what the normal amyloid beta peptide should look like. It's alpha helical structure, but it can form these beta sheet structures.
And then can you correct a misfolded protein by just boiling it and then doing the beta mercap, the ethanol?
You can, but like certain, like especially if you include some sort of chaperone, like for beta amyloid, you can correct it. In fact, that's one of the ways, but it'll spontaneously form these amyloid fibrillars over time, like really quickly. And there are certain mutations that you can introduce that really drive it to this. So there's probably mutations also that are safe, but if you have hydrophobic residues, substitutions here, they'll tend to drive the cross beta sheet formation and then fall right out of solutions. So this is the summary of chapter 4. And it's only 3:05. I thought we were just going to be getting here at 3:05. Any questions about chapter 4? So now we'll just jump to chapter 5.
I'm sorry.
Huh?
It was back when you were talking about X-ray crystallography, and you said that the cons for it is it's difficult with membrane proteins because it needs a hydrophobic solvent.
Well, membrane proteins are only going to be folded properly if they're in some sort of hydrophobic environment, at least the transmembrane domain, right? Because a lot of times aqueous or globular proteins, they'll have hydrophilic residues that project outwards into the solvent to interact with the water. Whereas a membrane protein typically has hydrophobic residues that project out into the membrane. Okay, and then they'll have hydrophilic, like if you're forming an ion channel. A lot of ion channels are just alpha helices, and they're typically pentamers. And if you're looking down through the channel like this, anybody tell me what kind of alpha helices these will most likely form? There's one word for it. Yeah, amphipathic helix. So they have residues that projecting outwards are going to be hydrophobic, and then the ones that project towards the center of the channel are going to be hydrophilic because you can form these water channels or ion channels. And then extracellularly, you'll have these large structures that'll bind your ligand that'll cause the shift in how these are arranged and open up the pore a little bit. So, but yeah, it's the important thing is they're amphipathic. So to crystallize those, it'd be difficult because you'd need some kind of aqueous environment. Plus, like instead of lipids, have some sort of organic solvent that you'll be able to maybe form reverse micelles. Have you heard of those? I guess you could maybe do it making reverse micelles and then evaporate the organic solvent off without denaturing your proteins. It'd be hard to crystallize. So, which is why the lab I used to do would look at these structures. They'd actually look at the nicotinic receptors in the nerves. And they were studying, you know, how binding the anesthesia affects those. And they'd make micelles and just use the transmembrane domains and look at those because they'd be small enough where the spin was correct with the size of the micelles. So, is that better? So were we on chapter 5? So now we're going to talk about the function of globular proteins. What did I do? I don't know what it did. There's a back arrow though. So reversible binding of ligands is essential for protein function. Anybody ever heard of the law of mass action? Do you remember that from general chemistry? Did they study that in general chemistry? Yeah. And then specificity of ligands and the binding sites. That's one of the things with proteins is they have high specificity. Ligand binding is often coupled with a conformational change. So what do we, did I explain what a conformational change is? Can somebody explain it to me? No?
Upon binding to a specific ligand, the change in the structure.
Yeah, so there's a three-dimensional change in the structure. It doesn't necessarily have to be upon binding to the ligand, but proteins, they're three-dimensional structures and there's movement. Sometimes binding to a ligand will induce a conformational change, and we're going to talk about that. It's an induced fit. So you induce this conformational change, which then may alter its affinity towards some other ligand, or alter the affinity of a neighboring subunit. In multi-subunit proteins, conformational changes in one, which is what we were just talking about, can affect the others, and that's called cooperativity. And you'll see in this chapter we're going to talk about positive cooperativity and how hemoglobin helps deliver blood to the tissues. Interactions can be regulated, and that's allosteric mechanisms that you see with a lot of these multi-subunit proteins. And we're going to illustrate it with hemoglobin, antibodies, and muscle contraction also. So functions of globular proteins, there's many. Storage of ions and molecules, it's myoglobin. What does that store? Yeah, it stores oxygen. Ferritin stores iron. Transport of ions or molecules, hemoglobin transports oxygen and
Hemoglobin.
CO2, right. Serotonin transporter also is another example. These are all globular proteins. Defense against pathogens, so antibodies. Who's in immunology? A lot of you guys. When is the first test in there?
Monday.
Next Monday? Okay. Just want to make sure it's not the same day as my first test. So defense against pathogens, antibodies, and cytokines, muscle contraction, actin and myosin. Do you guys know how muscles contract?
Sliding filament theory.
Huh?
The sliding filament theory.
Yeah, but why does that work? Do you know?
Yeah, it's a ratchet.
Spot and playing.
Yeah, we'll talk about that more detailed. Huh? What role does calcium play?
Oh, it opens the channel.
No, it's the depolarization that opens the channels, that releases the calcium, that then binds to the... Huh?
Isn't it troponin or tropomyosin, the calcium?
So it binds to the troponin C subunit of troponin, or tropomyosin. I forget all the words. But I got a little, what do you call it, animation I put on here that I like. And then biological catalysis, so chromatrypsin and lysozyme, those are 2 enzymes. So all of those are functions of globular proteins. And interaction with other molecules, this is basically going to be the law of mass, what did I say before? Not mass action, right? So a molecule that binds to a protein is called a ligand. So the molecule that's binding is the ligand. The region of the protein where the ligand binds is called the binding site, or the protein could also be called the receptor, right? And ligands bind via the same non-covalent forces that dictate protein structure. So why is non-covalent binding forces important for the binding?
Because they're weak.
Yeah, which means that they're transient and reversible reactions, right? Because actually, some enzyme inhibitors, there's a certain class that forms covalent interactions with the enzyme, basically kills the enzyme, right? So binding is a quantitative description of binding. You can think of it like this. You have a protein, or the receptor, plus the ligand. There's a rate constant that's going to govern how fast the 2 come together. And then this will dissociate also, so this is the Ka, or K of association, the rate, little Ka association. And then there's a Kd for dissociation, and they'll go back. So you can calculate both, the forward rate and the reverse rate via the rate laws, right? Do they do that here? Not really, but sort of. But they just show you what happens. This is the rate law of association. And that when that's equal to the rate law of dissociation, what is that called? Yeah, so the rate of association and the rate of dissociation are equal. That's when you're in equilibrium state. And you can solve and basically solve for all the concentrations. And that's going to equal the rate constants, which is all the constants. So you can make that your K, big Ka, not your, although it's the same thing as a Ka, but we're not talking acid-base reactions. It's the K of association, so the rate of association. And normally though, when we're talking about ligand binding interactions, you always hear Kd, right? Have you heard of a Kd before? That's the dissociation rate constant, which is just this inverted. So, and you can analysis in terms of the bound fraction. Have you seen this equation before? So rather than saying, certain concentration is bound, you can say percent fraction is bound. So they solve it like this. The bound fraction is going to be your ligand protein concentration divided by the total amount of protein there, right? So you have your bound protein and your free protein. So the fraction that's bound is going to be the bound protein divided by the total protein, right? But it's hard to measure this PL on its own, so it's better to have things as far as their constants. So you can solve for PL, right? And substitute the Ka times concentration of ligand times the concentration of P for the PL, and you end up with something that looks like this. And then if you get rid of all the P's, because you see how you can pull the P out here, and this P will divide. Do you want me to write that out mathematically? Because they don't show you in these PowerPoint slides, and I don't know what I did with the eraser. Did you see? Was there one here before? It's way back there. Can't walk. I'm lazy. So, oh, Thank you, sir. So where do you want me to show you this derivation from? From the very beginning, or?
Sure.
So basically, right, so total protein, and again, I apologize for my handwriting, it's pretty despicable, is going to be equal to the sum of all the protein that's there, right? So it's going to be equal to the protein that's involved with forming a protein ligand complex plus all the free protein in concentration. That's going to equal your total protein, right? So if you want to know the fraction that's bound is just going to be, and they give it a little, is that a theta? Oh, Greek letters. So theta equals bound protein divided by total protein, which is going to be equal to again that little figure, right? So then we don't want PL in there because we never know, unless we know the KAs and everything, exactly what PL is going to be. So we just want to have everything as L and P specifically, because when you do this, you can just add, you know, the concentration of L and the concentration of P that you're adding to something. And what was the equation? So Ka equals the concentration is it L? So yeah, it should be the products divided by the reactants, right? So Ka, so the products are going to be PL, right? And the reactants are going to be free L and free P. So if we solve for PL, you get, not equals, but we're going to have PL equals, and we just cross multiply, so Ka, or the association rate constant, or the equilibrium constant times ligand concentration times protein concentration, and then we'll substitute that in for PL here, and we get Ka times L times P divided by Ka times L times P plus P. So we're going to pull P out of this, right? So we get concentration of protein times the Ka times the ligand plus 1, right? Do you see how you get the 1 there? Because P, multiply P times Ka times L, you get that, plus 1, you get that. Do you see that? My handwriting is terrible. There should be an L in here. Okay. And then so then these P's, we can get rid of the protein concentration out of there all the way. And then how they get this is they basically divide this whole feature, if you multiply it by basically you pull the Ka out of here. Wait. Yeah, because we want to get rid of the Ka here. So we're doing the same thing. We're going to pull the Ka out. I don't know. probably confusing. So if we want to pull the Ka out of this, you'll end up with Ka times L plus 1 over Ka, right? And then this Ka can get crossed out with that. And all we're left with now are ligand concentrations and then 1 over Ka in the bottom. And do you guys remember what 1 over Ka would be equal to? Ka is the association equilibrium constant. 1 over Ka is the dissociation rate constant. So this is what binding looks like. So the fraction bound sites, so they have fraction bound, goes from 0 to 1. You're not going to have more than 100% bound. Depends on the free ligand concentration and the Kd. So as you increase, ligand concentration is known typically when you do these experiments. So as you increase, it'll go up in a log linear fashion, and then it'll asymptotically approach 100% bound. And the Kd can be determined graphically via the least squares regression, basically. And Kd is actually the concentration, equals the concentration at which 50% of the sites are bound. And this shows you oxygen, an example of a plot showing the oxygen binding to myoglobin, and it shows that same sort of goes up and then over. So when ligand binding is a gas, rather than saying concentration, it's the partial pressure of oxygen, which you guys will see. I think some of you saw it today, blood gases. So the interaction strength can be expressed as an association binding constant or per molar, but typically you always see the dissociation constant, and those units are going to be molar, right? Because you have, this would be per molar because this is in molar, this is in molar, and this is in molar. So these would cancel out, and you're left per molar for the Ka. Kd is flipped, so it's molar. And then the interaction free energy also is delta G. This is your standard state delta G is kilojoules per mole. And you guys remember this, right? Standard state free energy equals the standard state change of enthalpy minus the temperature times the change in entropy. And this is your Ka, Kd is the inverse of that. And the relationships between free energy and the Ka can be written like this. Free energy change equals negative RT times the natural log of Ka, or RT natural log, negative natural log of Ka, or the natural log of Kd. And this is what people always get confused about, are the magnitudes. So the smaller the Kd, the stronger the binding. Weak binding is when the Kd is larger, okay? So strong binding below 10 nanomolar, weak bindings greater than 10 micromolar. So, and then I think there's some examples here of different ligand and the binding strength. Anybody worked with avidin before or know what avidin is? So avidin has one of the highest affinities for its substrate that there is. Its substrate is biotin, and it's the molecule that carries biotin. You can extract it from egg whites like they do here, and its Kd is 1 to the minus 1 times 10 to the minus 15 power. So that's, how many molar is that? 1 times 10 to the minus 15? Huh? So what's it? 10 to the 6th is micro. 10 to the minus 6 would be micro, right? 10 to the minus 9th is nano. 12 is pico. So what is 15? Femtomolar. So that's femtomolar binding. So that's really strong. Whereas insulin receptors, 1 to the minus 10 to the 10. Antibodies typically around 10 to the minus 10 and so forth. So that biotin, avidin is one of the really strong binding reagents. And it's easy to add biotin to reagents. It'll just, you'll see in the future, forms a covalent bond with lysine on proteins. And you have this big biotin on a chain. And you can label things with avidin and you create a good reagent. A lot of times they'll use avidin biotin interactions for immunohistochemistry or ELISAs and things like that. So specificity, who's heard of that lock and key model of binding? Where it's specific and it's like that one key only binds into there and the proteins aren't going to change shape at all. They just have this, they just fit perfectly together and bind together perfectly. And so they're complementary in their size, shape, charge, or hydrophobicity and hydrophilic character. And it's called a lock and key model because they just fit perfectly together like a key fits into its perspective lock. This is an induced fit model, which I think you'll see more often than the lock and key model, especially for enzymes and proteins. They're going to alter their conformation upon binding, which will alter their function, typically. So this is an adaptation to the lock and key, and it's called the induced fit, and it allows for tighter binding of the ligand. Induced fit allows for high affinity for different ligands also. So both ligand and protein can change their conformations once they bind together. And then with this new conformation, it may increase their, turn on some sort of specific enzymatic activity or alter its affinity in some kind of regulatory fashion to alter cellular functions. So globulins are oxygen binding proteins. Protein side chains lack an affinity for oxygen. So some transition metals bind oxygen well but would generate free radicals if they're free in solution. So you can't have the free transition metals going through your bloodstream because it'll cause oxidative damage. Organometallic compounds such as heme are more suitable, but the F2 in free heme could be oxidized to the ferrous form. And the solution is you capture the oxygen molecule with the heme that is protein bound. And myoglobin is the main oxygen storage protein, and hemoglobin is the circulating oxygen binding protein. And this is the structure of the heme group. So you have all these nitrogens. Iron can form 6 coordinate bonds. This square planar is where they'll accept the electrons around the iron. So the iron was coordinated with the heme group. And then you have 2 other sites that are 180 degrees apart. One that will coordinate with an oxygen, and the other actually forms a bond with the histidine on either the hemoglobin or the myoglobin. And this shows you a picture of that. Histidine bound to the iron, bound to an oxygen. And actually, when the oxygen isn't there, this histidine pulls this heme group out of planarity. So it's typically not planar when the oxygen's not bound, but when it's bound, it's planar. Oh, at least it is for hemoglobin. I'm not 100% sure for myoglobin. So binding of carbon monoxide. So carbon monoxide has a similar size and shape to oxygen, and it can fit into the same binding site. And it is over 20,000 times better binding than oxygen to myoglobin. And it's because it only has the one lone pair at the end of that carbon. Because remember, carbon monoxide structure is what, like this? So it's like got a triple bond, right? And then partial charge, because this is, I forget the Lewis structures. You guys good at those?
No, full charge.
Huh?
Because it's a triple bond.
So yeah, you would have like, so you can have, which this will be then a positive charge, right? And then that would be neutrally charged. This will fall down. No, I'm not going to do, I haven't done Lewis structures in years. Since the last, you were probably there the last time I did Lewis structures.
Yeah.
So, anyways, the protein pocket that surrounds the heme group is going to decrease the affinity for carbon monoxide, but it's still able to bind about 250 times better. And carbon monoxide is highly toxic as it competes with the oxygen. One of the reasons is because it forms, you know, it's basically 180 degrees. Get the lone pair there for the carbon, and you have 180 degrees that'll coordinate with the iron. Versus the oxygen, those lone pairs are off to the side, right? And this shows you how the histidine will actually stabilize that oxygen in that configuration. Histidine, and this is part of the myoglobin chain. Again, this is what the binding affinity for myoglobin looks like, and could it be used as a transport molecule? If you look, so in the lungs, the pressure is about 13 kilopascals, so certainly could get saturated with oxygen in the lungs. But the partial pressure of oxygen in the tissues is only about 4. So it's basically, myoglobin still going to be saturated in the tissues, right? So it wouldn't be able to drop off any of that oxygen it picked up in the lungs to the tissues where it would be needed for metabolism. So if you lowered the affinity of myoglobin, it could help, right? Because if you lower the affinity, this won't get saturated until much later. Does that make sense? It'll shift that curve basically to the left. Sort of like this, a lower affinity state, and then this is the high affinity state. And this is actually what you kind of see with hemoglobin. So hemoglobin will have 2 different conformations. One's in a low affinity state, one's in a high affinity state. And you pretty much, when there's no oxygen bound to hemoglobin at all, it typically has a low affinity state. But as you bind oxygen, it steers more to this high affinity state. And you end up with this curve that looks somewhat in between. As you increase the partial pressure of oxygen, you increase the affinity for the oxygen that the hemoglobin has, and as oxygen levels drop off, you decrease the affinity of the oxygen, so you're able to dump off more of that oxygen where there's less oxygen around. So how can affinity to oxygen change? And this happens is because the protein has to have multiple binding sites. So binding to one subunit, if you guys remember, hemoglobin contains 4 subunits. When oxygen binds to one, it alters the affinity for all the other subunits to increase its affinity for oxygen. And that's what's known as positive cooperativity, where the first binding event increases the affinity at the remaining sites. And you'll recognize that sigmoidal binding curve. Like we saw here, it's kind of like this S shape where it starts off low and then it increases and then levels off again, like a normal binding curve would look. Negative cooperativity, you might see, that's the first binding event, reduces the affinity at the other sites, but hemoglobin has positive cooperativity. And it's going to do that because something like this, you get cooperativity, the binding site, with no ligand, you got these pink segments that are flexible. Few conformations facilitate ligand binding, and green segments are stable in the low affinity state. However, when you bind one to the other, it causes the other subunit that's not bound to be more stable with a higher affinity state, and then it'll be able to pick up its ligand also. So typically cooperative proteins, oh man, are we late now? 8?
Isn't it 340?
Is it?
Yeah.
Oh, it is. Why did I think 330? Because I'm stupid. Okay, so cooperativity. Cooperative proteins have to have multiple ligand binding sites, so the K of association becomes this, where you have multiple sites. You include N for the number of sites that you have. Theta is going to be equal to L to the N divided by L to the N plus the KD. Taking the log of both sides gives this Hill equation. Have you guys ever seen the Hill equation? So it's the log of basically the fraction bound divided by the total, which equals N log of the ligand minus the log of the KD. And the Hill coefficient, which is this, is the degree of cooperativity that you have. And N equaling 1 equals no cooperativity, basically. That's cut off for some reason. And this is like graphs of the Hill plot. And you can see the two different binding states that you'll have with hemoglobin. And if you compare those to myoglobin, there's no cooperativity, whereas hemoglobin has a low affinity state and then a high affinity state. And the two models of cooperativity, including concerted or sequential. Concerted is when one ligand binds, boom, they're all in a high affinity state and they all switch their conformation. Whereas sequential is when one, the next one will switch it switches its state, switching the state of the next one, and then the next one, and the next one, and so forth. So different models between the two. And cooperativity is a special case of allosteric regulation. And you'll see allosterism is really important in biochemistry. So that's how most of metabolism is regulated, via allosteric interactions. So allosteric protein is binding of a ligand to one site affects the binding or properties of a different site on the same protein. It can be both positive and negative. And homotropic is where the normal ligand of the protein is the allosteric regulator. Oxygen is a homotropic regulator of hemoglobin, right? Heterotrophic is a different ligand affects the binding of the normal ligand. 2,3-BPG or 2,3-bisphosphoglycerate is a heterotrophic regulator of hemoglobin binding to oxygen. And we'll talk more about that. And that's really important when we're talking about delivering oxygen to the fetus. Fetal hemoglobin doesn't bind to 2,3-bisphosphoglycerate very well. And then cooperativity equals positive homotropic regulation for the most part. So hemoglobin binds oxygen cooperatively. Each subunit is similar to myoglobin. However, they form a quaternary structure with 4 subunits, versus myoglobin is just a monomer and it doesn't have any cooperativity at all. It just stores the oxygen. But with the hemoglobin, we have to not just grab the oxygen, we got to be able to dump it off where it needs to be. These are structural similarities between hemoglobin and myoglobin, which indicates probably hemoglobin evolved from myoglobin years ago. This is the overall structure of subunit interaction between hemoglobin. You have 2 alpha subunits and 2 beta subunits. And they have the interacting areas highlighted like that. And more details on the interaction. For example, you have a lysine on the C5 of the alpha subunit interacts with the histidine on the beta subunit. So why would lysine and histidine interact? So histidine can almost be either acid or acidic or basic. Its pKa is right just below 7. And then the lysine is going to be positively charged, so the histidine can then coordinate that. It'll hydrogen, kind of try to pull that charge away from the lysine. Then you have aspartic acid, which is So this is the histidine. So that's coordinating with the aspartic acid and then forming a salt bridge with that lysine, that is. And this shows you the individual subunit interactions. And when we're talking allosterism, typically there's this model where the protein can be in what's known as a tense state or the T state, or the relaxed state or the R state. So the tense state has more interactions and it's more stable and it has a lower affinity for oxygen. So tense state, it's more interactions between subunits. Lower affinity for oxygen, or a relaxed state, it's fewer interactions, it's more flexible and has a higher affinity for oxygen. Once you get oxygen binding, which it'll trigger the tense to an R conformation will change and the other subunits will have that relaxed state conformation. Conformation change from the T state to the R state involves breaking ion pairs between the alpha 1 and beta 2 interface, and that would be your lysine and histidine and so forth, or lysine and aspartic acid. So R states and T states of this is your T state, low affinity for oxygen. It looks like you have histidine here interacting. And then for the R state, the histidines are where? They're like down here, not interacting with their respective subunits that they are in the T state or the tense state. So you also get a conformational change that's triggered by the oxygen binding. And what happens, this is what I was saying before, with no oxygen bound in the heme, it's not planar. And the histidine on the F helix kind of coordinates the iron. When oxygen binds, it helps pull this into planarity, which will pull this whole alpha helix a certain distance and help change the overall conformation of the quaternary structure of hemoglobin.